Let's talk about meiosis specifically, because there's some really important things that are happening during meiosis. So, most of the time, your cell just has the DNA hanging out inside of it. It's in interphase. Your cell spends most of its time in interphase performing cell functions, and most of the time that's where your cell is. But when your cell needs to divide, it goes into cell division, right? And it needs to copy this DNA. And your DNA is a very long molecule, even though it's tucked away in the nucleus, and the nucleus is a microscopic organelle. If you took your DNA out and stretched it, it would be, and just not pulled it, but just made it straight, it would be about the length of a car. So you have all of that genetic material coiled up very tightly inside of the nucleus of the nucleus of a cell. It's amazing. So when your cell has to divide, it has to make sure that it divides all of the information that's on the DNA. And it's evolved an excellent process to do this. It first copies itself. And I'm going to do that right now. Prophase. Prophase 1. The DNA has duplicated itself and has now formed these beautiful X-shaped chromosomes. Here's a chromosome, and here's a chromosome. Chromosomes, chromosomes take that beautiful X shape, okay? In prophase 1, we call these chromosomes homologous. Homo meaning same. And we call them homologous chromosomes because, I'm going to put them over for you, they are carrying the same information, just two different versions of them. One version from one parent and one version from the other parent. But it's the same information. It's your eye color, the type of hair, your dimples or no dimples, earlobes, all of your information, okay? So these are called homologous because they're carrying the same information. Now, each chromosome is composed of what's called a sister chromatid, right? So here's one sister chromatid. And here's the other. This one, again, has one sister chromatid, two sister chromatid. Each chromosome is composed of two sister chromatids. So if I'm looking at this right here, how many sister chromatids do I have all together? I have 1, 2, 3, 4. So that is why this is called a tetrad, because tetrad means 4. Okay, so homologous chromosomes are forming a tetrad. All right, now they do something really remarkable. They are touching each other. Sometimes they really intertwine with each other. And what happens when they do this is they actually share genetic information. So maybe the alleles for brown hair and brown eyes that were from one parent are now going to be on this chromosome, and these will be on that chromosome. Maybe we're going to get these two alleles. Gee, Legos are hard. Maybe you're going to get those two chromosomes that are going to be on that. I mean, those two alleles that are going to switch with it could be anything. It's random. You never know. This, when they touch and they kind of cross each other, this event is called crossing over. And it is an essential event to know and understand because this creates completely genetically unique, offspring. Never before has this chromosome had this set of genes, and never before has this chromosome had that set of genes. It's due to crossing over, and it creates completely, did I spell that right? I might not have. It's okay. Completely unique. How about if we actually write that so it's in frame? How about we do that? So crossing over creates completely unique offspring. It is one of the reasons why my sister has fair skin, blue eyes, and freckles, and I have olive skin, brown eyes, and hardly any freckles at all unless I'm out in the sun for a long period of time. This is one of the very important, I just can't stress it enough. I don't even, what should I circle it in? You have to understand that this is an essential process of meiosis, because we're going to talk about this when we talk about evolution, because evolution only happens because there's genetic diversity in individuals. Okay, so that is prophase 1. Now, what happens next? We go into metaphase 1. Okay, so here we are during metaphase 1. Our 2 chromosomes, and by the way, we have 2 chromosomes, right? 1, 2. They've lined up in the center of the cell. So our diploid number is 2. We could also say that we are 2n equals 2, because diploid and 2n mean the same thing. Okay? All right, now, it is completely random how these alleles are going to line up. Okay? There's no rule. So, they could line up like this, they could line up like that. We don't know. They're going to assort independently. That is another very crucial step in meiosis, that alleles will assort independently. Independently. This is another way that we achieve genetic diversity. Another way that sexually reproducing organisms, I'll just say sexual reproducers. Let me move it over. Sexual reproducers get genetically diverse young. And I wrote it in red, so you know it's really important. If you don't have genetically diverse young, your species won't evolve. You need genetic diversity within your species in order to evolve. So, independent assortment is a way to get genetic diversity, and in the previous, we talked about crossing over is a way to get diversity, and both of these 2 things happen during the process of meiosis. You really see independent assortment when you do dihybrid crosses, and we'll do that in a subsequent video. Okay, so they've lined up in the center. You have centrioles right here that are going to, I shouldn't have done two on them. You have centrioles right here that are going to attach to the center of this chromosome and pull them to opposite sides of the cell. That is what our next phase is, which is anaphase. So I'll elongate my cell a little bit. There. That is anaphase 1. Still one cell. These moving. The cell is getting longer. Until finally we reach the end of stage 1 of meiosis. Stage 1 of meiosis. At which time we have 2 cells. So we have gone through telophase 1 and cytokinesis 1. And how many chromosomes do I have in this cell now? The parent cell I had 2. How many are here now? That's correct. There is only 1 chromosome. So we are not 2n equals 2. We are now n equals 1. We are now n equals 1. You could have figured that out by just doing the simple math. Well, if I was 2n equals 2, then when I get to telophase, I should be n equals 1. You can figure that out yourself to make sure you're doing it correctly. So, we're not done yet though. We now have to move on. Both of these cells are no longer diploid because they only have half the chromosome number their parent did. Half and completely unique chromosomes. So both of these cells are now haploid. Both are haploid. Okay. Now we go into stage 2 of meiosis. Now we have 2 cells going through it. There. Prophase 2. Now we move on to which phase? I know, you're stumped. Are you ready? Two cells going into metaphase 2. Now, we're going to have the centriole grabbing that part of the chromosome, and the centriole pulling at that part, and this one pulling at this part. So what's going to happen are these two sister chromatids are going to now separate. Right? The second stage of meiosis looks a lot like mitosis in what the chromosome goes through, except these are completely unique chromosomes, and in mitosis, you're creating clones of the parent cell. There's nothing unique about them. They're exactly the same type as the parent, or they carry the exact same information as the parent in mitosis. In meiosis, these are completely unique genetic combinations. Okay, so now on to anaphase 2. And in anaphase 2, we have the chromosome now completely separating, has split apart, completely separating itself So now we're back to two separate sister chromatids, right? And then the next phase, how many cells will we have individually? In telophase 2 and cytokinesis 2, we should have we now have 4, 1, 2, 3, 4. They're all n equals 1. So they're all, what do we call them when they have half the number of chromosomes as their parent? They're all haploid. All are haploid. Right? Because they have half the number of chromosomes as their parent. And all are genetically unique. And how did this genetic uniqueness occur? What two things? We had crossover and we had independent assortment. And both of those events led to these genetically unique, never seen before combinations of chromosomes or genes. Okay? Combinations of genes would probably be a better way to phrase it. These are all gametes. So if you're a male, these are what your sperm would be. You've just created sperm. If you're a female, you've just created eggs with half the number of chromosomes. So when the sperm fertilizes the egg, you have the right number of chromosomes. Okay? That's meiosis. All right, awesome.